This lecture is a brief review of the main concepts in parts 1 and 2 of MCDB 101B, Eukaryotic Genetics. Let us not forget our fundamental law of genetics. Phenotype equals genotype plus environment, as well as our more quantitative formulation of this law from part 3. The total phenotypic variance in a population equals the genotypic variance, which is the variance due to different genotypes, plus the environmental variance, which is the variance due to different environments. Gene, allele, genotype, phenotype, heterozygous, homozygous, dominant, recessive, true breeding, trait, these are terms that you are familiar with now. You understand the basics of a cross, starting with the parentals that give rise to the F1 progeny, which when self or intercrossed, give rise to the F2 progeny. To calculate the genotypic and phenotypic ratios, we first determine the gamete genotypes from each parent using Mendel's first and second law, when appropriate. Note the violations of the Mendel's law, especially of Mendel's second law, when the two genes are linked. Then we put the gamete genotypes on each side of the Punnett square and fill in the square, generating the progeny genotypes. We then interpret the genotypes into phenotypes. A test cross to the homozygous recessive can be used to determine the genotype of an individual showing the dominant trait, i.e. whether the individual is homozygous or heterozygous. Classical Mendelian genetics forms the foundation of modern genetics, but it applies to very few traits. Most traits are complex traits that violate Mendel's laws or assumptions. In particular, you need to understand concepts such as incomplete dominance and codominance, and more importantly, concepts like incomplete penetrance, when a given genotype does not always show the expected phenotype, and viral expressivity, when the expected phenotype can be expressed at different degrees. Remember our example of the purple peas and the different shades of purple, which represent variable expressivity. And incomplete penetrance is when we expect a purple pea, but sometimes the progeny are the white peas. Then we move on to epistasis, which is the idea that more than one gene may determine a trait. And in particular, we focus on two genes interacting to determine a trait. And our first example is complementation, which is a special type of epistasis. And here you want to remember the purple and white flowers. The idea is we have two different genes that determine whether the flower is purple or white, but you can have two different true breeding white flowers, white 1 and white 2, and when you cross them, you get a purple flower. And the reason why is because you have two genes that are necessary to make the purple pigment, and one white flower is homozygous recessive for one gene, and the other white flower is homozygous recessive for the other gene, and when you cross them, you get the double heterozygote, which can go ahead and make the purple pigment. More broadly, as I mentioned, epistasis is when you have more than one gene, and in our case we focus on two genes interacting to determine a trait. And so remember table 3.2 and our different examples of epistasis. And again, just like on midterm 1, you don't have to memorize these examples. I'll give you, for example, the pathway, and then you can do the cross, fill out the Punnett square, then based on the pathway, interpret the genotypes into phenotypes where you have more than one gene involved in determining the phenotype. And table 3.2 is a good summary of epistasis. Humans, we don't cross humans, and so to determine the mode of inheritance, we look at pedigrees. And so there are two main types of questions that I'll ask. One is to determine the mode of inheritance from the pedigree, and the second is to rule out the mode of one or more modes of inheritance from a pedigree. In addition, you want to be able to infer genotypes from within the pedigree depending on the mode of inheritance. There's also the genetics of genes on the X chromosome, the X-linked or sex-linked inheritance. It's important to know that, as well as the concept of X chromosome inactivation, which is the idea that in women with two X chromosomes, in all somatic cells, one of the two X chromosomes, and it's randomly chosen, is inactivated. And so only one of the X chromosomes is expressed, and the calico cat was a good example of X chromosome inactivation. Meiosis is critical. It's important to understand meiosis. Meiosis is how you go from a diploid cell to generate your haploid gametes. So understand the steps through meiosis 1, through meiosis 2, as well as when you have non-disjunction in meiosis 1 and non-disjunction in meiosis 2, and the differences between mitosis and meiosis. Finally, there's the distinction between germ cells and somatic cells. The germ cells give rise to the gametes, so they are the cells that give rise to the gametes as well as the gametes, and the somatic cells are all the other cells in the body. Moving on to part 2 of the course, we focused on the concept of linked versus unlinked genes, where you have two genes, and if they're on different chromosomes, then Mendel's second law applies and they're unlinked. Or if they're on the same chromosome and far apart, then they still act as if they're unlinked. But if they're close together, then they're linked, and they tend to segregate together. So we have the idea of parental and recombinant types. So it matters how the alleles are paired on a given chromosome. And think about the different progeny groups, whether they're parental or recombinant. And then you can get recombination between alleles on the same chromosome. And we can calculate the recombination frequency by looking at the number of recombinant types over the total number of progeny. And the way we do that for a 2-point cross is through making a dihybrid cross, or in a 3-point cross by making the trihybrid cross. And from those we can calculate the recombination frequency, and we can use recombination frequency to calculate map distances and to make genetic maps. And remember, the recombination frequency is always less than or equal to 50%. So some of the things you should be able to do from 2-point and 3-point cross data is calculate map distances from the cross, figure out the map order, and in particular the gene in the middle. And the key with the 3-point cross, and the advantage of the 3-point cross for the 2-point cross, is the ability to count the double crossovers. And so you must include those in the calculation. And you have to go also, starting from a map, back to the progeny count. So you go in both directions. From the progeny counts, we can calculate map distances, and from the map distance, we can infer what the progeny counts are. And finally, there's the concept of chromosomal interference, which is the idea that you don't observe as many double crossover events as you expect from the individual recombination frequencies in region 1 and region 2. Then we covered the concept of chi-square, and remember our example of flipping the coin. And the idea of chi-squared is whether the observed data fits the expected data, in particular an expected ratio. The null hypothesis is that we expect a certain ratio, and in the case of the coin it was a 1-to-1 ratio for a fair coin, and in the case of linkage or not, the null hypothesis is that the two genes are unlinked, and hence we expect to see the same number of a 1-to-1 ratio of parental to recombinant types. And then we calculate the chi-square value for the hypothesis. So again, this calculation involves subtracting the observed data from the expected data, and I'll provide the formula on the back page of the exam. And then we use a chi-square. Once we calculate the chi-square value, we can use a chi-square table to calculate the p-value, depending on the number of degrees of freedom, where the number of degrees of freedom is the number of classes minus 1. And then from that, from the p-value, we can reject the null hypothesis if the p-value is less than 0.05, and we can reject that at a confidence level of 95%. The next topic is chromosome abnormalities. Remember, there are different abnormalities: deletions, translocations, inversions, duplications, and extra and missing chromosomes. And in particular, aneuploidy is when instead of the normal pair of homologous chromosomes, you either have an extra, so trisomy, or you're missing a chromosome, or monosomy. and trisomy and monosomy and aneuploidy arise through non-disjunction during meiosis. And remember, non-disjunction in meiosis 1 and non-disjunction in meiosis 2. The central dogma from DNA to RNA protein really encapsulates how we go from the genotype to the phenotype. The genotype is in the DNA, that DNA is transcribed into RNA, and that RNA is spliced and then translated into protein, and it's the protein that does the activities in the cell that results in the phenotype. So first, the step from DNA to RNA is transcription, and remember all the different components of transcription: the promoter, the enhancer, silencer, RNA polymerase, the transcription factors, which can either be transcriptional activators or transcriptional repressors. Then once the primary transcript is made from the DNA template, it gets spliced, and so only the exons are kept while the introns are spliced out. And that results in the mature mRNA. And remember, it's the spliceosome, which is the enzymatic machinery that does the splicing, and the GUAG rule is how the spliceosome figures out where the introns are to splice them all out. Then alternative splicing is how in different cells you can give rise to different proteins depending on which exons are preserved as well as which introns are spliced out. And so in different cells you can imagine different introns are spliced out and you can produce different proteins in different cells from the same gene. Gene expression captures the idea that different cells have the same DNA but different proteins. So how do you generate different proteins from the same DNA, from the same genotype? Well, the regulation can be at any one of the levels, transcription, splicing, or translation, but it mainly occurs at the transcriptional level. And so what's the difference between a neuron and a liver cell? Well, the neuron has neuron-specific transcription factors which transcribe neuron-specific genes which make neuron-specific proteins. And then the liver cell expresses liver-specific mRNAs that get translated into liver-specific protein. So for translation, the mRNA has an open reading frame, and the sequence on the mRNA is read 3 nucleotides at a time, which is the codon. And it starts at an initiator codon, which is the AUG, which encodes for methionine, and translates 1 codon at a time to the terminator codon where it stops. And that, from the initiator codon to the terminator codon, is the open reading frame. Because the codon is 3 nucleotides long, that can encode for 64 different codons, 4 times 4 times 4, but there are only 20 amino acids. So the genetic code is degenerate, meaning that more than 1 codon specifies the same amino acid. And the way that degenerate code is implemented is through wobble base pairing, typically through the 3rd position in the codon. The genetic code is, in a sense, specified by the tRNA and the tRNA synthetases, and the tRNA synthetase is the one that adds the amino acid to the tRNA, and the tRNA has the anticodon which binds the codon. And make sure you understand the concept of reading frame and how that reading frame can be shifted by a frame shift mutation, which is an insertion or a deletion that shifts the frame so that you're no longer in the proper frame when you're translating the codons. And so you have a 1 base pair insertion or a 2 base pair insertion or a 1 base pair deletion or a 2 base pair deletion. You shift the frame, whereas a 3 base pair insertion will insert an extra codon, but after that the frame is maintained. Finally, make sure you understand the different types and classes of mutations. There's the silent, missense, nonsense, frame shift. Again, make sure you understand what those different types of mutations are. There's loss of function mutations, hypomorphic, null, haploinsufficiency. There's the gain of function mutations, the hypermorphic, neomorphic, antimorphic, and the concept of a dominant negative mutation. So that's important, the types and classes of mutations. A mutant screen is how we can identify experimentally what genes are involved in a process, and we characterize different mutants using a complementation test, which uses the concept of complementation to determine whether mutations are in the same gene or different genes. We went over how a mutant screen can help us determine what the pathway is. And there are different pathways. There's a protein pathway, which is that a protein can activate or inhibit another protein, and you can have a chain of those activation and inhibition steps. And the second type of pathway are the biochemical pathways, where you have the proteins and their enzymes, and they catalyze a reaction. So you have some substance that's converted into another substance, which, for example, can be converted into some dye that results in some phenotype, and the protein is catalyzing those particular reactions. And finally, a suppressor mutant is a mutation that suppresses an original mutation. So if you have an original mutant and then you add in a second suppressor mutant, the two together can restore the wild type phenotype. So the suppressor mutant restores the wild type phenotype from the original mutation. And make sure you understand maternal effect, mutation in gene, and mitochondrial inheritance, and how they're similar and different. And finally, epigenetic inheritance. What is epigenetic inheritance? And understand the distinction between epigenetic inheritance and genetic inheritance, in which the genetic inheritance, you have the change in actually the DNA sequence, which gives rise to a change in your genotype, whereas in the epigenetic inheritance, there's no change in the DNA sequence, but there is a preservation of whether a gene is expressed or not, and that's accomplished through DNA methylation and silencing. And understand the differences between paternal and maternal imprinting. And that concludes our summary of Parts 1 and Parts 2.